Hello, everyone, and a very warm welcome to Kognito Key. I'm so thrilled to have you here today. Let's just dive right into a truly fascinating topic, the maintenance and preservation of pure cultures. You can think of it as, well, a guide to microbial immortality. Today, we're exploring the incredible journey of isolating microscopic life and, more importantly, figuring out how we actually keep it alive for decades.
Now, to understand microbial immortality, we first need to get a grip on the concept of a pure culture. You see, in nature, our environment is just packed with a vastly mixed microbial population. Studying them effectively means we literally have to pull a single specific cell out of this crowded microscopic city. We do this by aseptically transferring that one cell to a fresh, nutrient-agar medium. We're isolating it so we can study its unique shape, size, and physiology without any interference from all the other microbes out there.
So let's look at part 1, the 5 forms of pure culture, and how we categorize microbial growth.
How do we actually grow them in the lab? Well, we've got 5 main types. First up is the streak culture, which isolates discrete, individual colonies by gently spreading a tiny bit of inoculum over the surface. Second, the stab culture. For this, we use an inoculating needle to literally stab the culture deep down into the middle of a solid medium. Third, we have the stock culture. And this one's all about subculturing to maintain the microbe's viability over time. Fourth is the starter culture. These are active organisms prepared specifically to kick off fermentation, like in industrial enzyme production. And finally, number 5, the enrichment culture, where we provide highly favorable growth conditions to isolate a very specific microbial cell of interest. Each one serves a really specific, powerful purpose in biotechnology.
But wait, how do we know our culture is actually pure before we lock it away for storage? Well, scientists use a strict checklist for this. To assess purity, all the isolated colonies have to look like identical twins. I mean, they need to have the exact same size, shape, texture, and color. Then, under the microscope after staining, they must look absolutely identical. And finally, they have to yield the exact same biochemical results. Only when it passes this rigorous test can we confidently call it a pure culture.
Which brings us to part 2, halting time. We're looking at the 4 methods of preservation.
You see, in the lab, contamination and cell death are the ultimate villains. If left alone, our microbes are just going to slowly use up all their nutrients and eventually die from starvation and toxic waste. So biopreservation techniques step in as our ultimate time-stopping solutions. The secret here is that slowing down metabolic activity can literally stretch time. First, we have refrigeration at 0 to 4 degrees Celsius. Now, this slows them down, but it only buys us about 2 to 3 weeks for bacteria. Next is paraffin coating. By vertically covering the microbial growth with mineral oil at room temperature, we trap the moisture, maintain anaerobic conditions, and preserve them for a solid 1 to 2 years. But if we want real longevity, we look at cryopreservation. Freezing them down to between -150 and -196 degrees Celsius keeps them viable for 10 to 30 years. And finally, lyophilization, which can remarkably preserve our cultures for more than 30 years. That's a long time.
Let's look a bit closer at cryopreservation. This is a highly effective technique that uses liquid nitrogen freezers to reach an incredibly cold -196 degrees Celsius. But here's the catch. You can't just freeze them directly. To freeze cells this cold, we absolutely have to use special cryopreservative agents like glycerol or dimethyl sulfoxide. Why? Because without them, deadly ice crystals would form and just completely shatter the cells from the inside out.
Now contrast that with lyophilization, which you might know as freeze-drying. This is where we freeze the culture and then dehydrate the sample through a vacuum process called sublimation. For this, we use lyoprotectants like sugar, literally sugar, to stabilize the cells. The best part is that it turns the culture into this tiny dry state that is incredibly space-saving. It makes it extremely cost-effective and easy to just pack these small vials and ship them to other research labs anywhere in the world.
Now I want to pause for a second and ask you a critical question. After 30 years of deep sleep, perfectly frozen in time in a tiny vial, how do we revive these dormant cultures? How do we wake these microbes back up?
Well, the answer is subculturing. Subculturing is simply the passaging of cells. We take a little bit of inoculum from that dormant, pure, old culture, and we carefully transfer it to fresh, nutrient agar slants. By introducing these sleeping cells to a brand new, fresh growth medium, we restart their life cycle, wake up their metabolism, and successfully propagate the cell line for our ongoing research.
And that brilliantly illustrates how biopreservation plays a pivotal role in maintaining cell integrity and functioning. Keeping these microscopic wonders safe ensures they're always ready for the next big scientific breakthrough. But it leaves us with a fascinating question. If we can successfully pause and restart life at the microscopic level for decades, what secrets might preservation hold for the future of complex biology? Thank you so much for watching. If you enjoyed this explainer, please hit that subscribe button for Kognito key, leave a like, and share it with everyone who loves science. See you next time.